So welcome back everyone to the second session of the second day of FDP. And, I thank Dr. Deepti for accepting our invitation to be a resource person and to deliver a talk in this session. For a brief introduction, Dr. Deepti Gangwar is currently working as a manager in Analytical Department of Biotechnology, Pharma, Gujarat. After qualifying CSIR NET in 2004, she has completed a PhD in biotechnology from International Center for Genetic Engineering and Biotechnology, JNU, in the year 2010, under the mentorship of Professor V.S. Chauhan. She has around 10 years of experience working in various domains of analytics and covering a wide range of pharma molecules, from small molecules to biopharmaceuticals. Her current profile caters to biosimilars, where she is working on quantification of host cell impurities using various analytical techniques. With this brief background, I invite Dr. Deepti to deliver a talk on host cell proteins determination as a part of process-related impurities. Over to you, Dr. Deepti. It is muted. Still, it is your mic is muted.
Am I audible now?
Yes.
Okay.
Yes.
So from where I'm supposed to share my screen, because I'm not able to see that option. So it becomes.
It is the fifth option after the hand.
Okay.
Option.
Present now. Says present.
Yes.
Your entire screen. Fine. So is it my screen is visible now?
Just not yet. I think after clicking that, you have to click that your entire screen.
Allow. Is it visible now?
No. Yeah, it's coming.
It's coming. Okay.
Yes.
It's there?
Yes, yes.
Am I audible? And is my screen visible? So
yes, yes.
All right. So thank you, Dr. Soma Sundaram, to give me this opportunity. And I hope I'm able to do justice with this platform. So I welcome everybody who is on board. So before actually I start, I would like to ask you a very small question, which might raise your interest in maybe what is coming next. So the question is, what do you think makes humans human? And I would really appreciate if people who are on board can, give me certain answers to this question. So, like, what do you really think have evolved us into the present day humans that we are? So, any answers from the people who are listening to this question?
Once again, could you please repeat the question?
The question that I have is, what makes humans human? That is us, I mean. What do you think has evolved us into the present day humans? I mean, what feature of us, what nature of us, I mean, what aspect of us has evolved us into the present day human?
I think there is some little bit break in your voice.
That becomes a problem actually. Hold on.
Yeah.
Am I audible now?
Yes, yes.
Okay, so I think I'm not very sure that whether people have listened to my question. So, okay, let me just move forward then. So, if somebody has to ask me that what I think, what aspect of us has made, us evolved into the present day humans, then I would really say that I particularly believe that it is the curiosity. Yes, I particularly think that it is curiosity, and because of this curious nature, humans have particularly explored everything that was around us for the betterment of human race. So, since before I actually move on to the topic, so there are certain small feelers that will connect you to the topic. So this actually dates back to the prehistoric age where we actually utilized everything around us for slowly evolving into what, our present day technology, our present day understanding about everything around us is. So this reminds me actually of Stone Age first in case where we started utilizing the like non-living things which were around
Your
us.
slides are not moving.
Hello?
Your slides are not moving. I think still it's in the first slide.
God. It's not moving yet.
No. I think while sharing, you have to share the entire screen and then you
I
have to
have
open.
shared that only. I have shared, just hold on.
The screen is visible, but it is like
It is
it's
like
not.
share. I am sharing my entire screen. Sharing
Yeah,
your
but it is
entire
not.
screen.
Your PPT is not in full screen. Yeah, now it's moving. I think now it's not in full screen mode.
Now it's okay.
Yeah. So like this only you have to move. I think from slide by slide, not making as full screen.
Uh, is it visible now? I mean, is it okay
The
now?
second slide now.
Yeah. So is it okay if I've moved like one click further? So is the
Yes.
caption visible?
Yes.
Okay, fine. So is this slide visible now?
Now second slide.
Now I have moved on to third one.
No, it's not because I think you made it into slideshow mode. Then it will not work. I think there is some option while sharing the PowerPoint.
Sharing or I have actually shared my entire screen, so I'm not very sure.
Okay.
This becomes a problem actually. So how do you want
Yes.
me to then
Yeah.
move
Now
forward?
it's going to third. Now third is visible. So you can
I
without
think it is
making it to full
slow.
screen.
That is the only problem. I think it is slow because even if I'm clicking, is it visible now? So then
Yeah.
I have moved.
Third slide.
Okay, the moment I'm making it as a slideshow, it is not. Then it
Yes.
becomes. You want me to log in and try it again once because otherwise it won't give you a feel
Yes.
of it.
Okay.
Okay, I'll just hold. So I will again click this. I am your entire screen. Okay, is it visible now? Third slide.
No, not yet. Else if you can share as a Google Drive or something, then I can also share from here. Yes, now it's fine.
Now it's fine.
Yes.
So I will just quickly scroll. So just let me know whether it is going fine. Yeah,
Yes. Go ahead. Yes.
fine.
Yes.
All right. So I will again start. Maybe then people who have missed it and the slides were not visible. So I actually began with asking this very simple question that what do you think makes humans human? And my answer to this question was curiosity. And when I said this, I actually mean to say that since prehistoric times, humans have actually started exploring everything around them out of their curiosity so that they can start getting information what is available, how we can utilize it for the betterment of human race. So I actually started saying that this reminds me of the tool age where we started utilizing non-living things around us and made them fit for purpose. While we were utilizing non-living things, we also started exploring the living things around us. So be it plants for the purpose of agriculture, or be it the animals, like when we started domesticating them or when we started using them as food. Not only at this particular level, we also started exploring in terms of what our how the tools can evolve, what are the various technologies that have evolved. And if you have ever gone across the history of science and technology, so humans have a rich history of around 4 billion years of those age, which have led us to the present day inventions. And now, as we all know, that we are even leveling it up with machine learning and artificial intelligence. So while this is all happening at a mega level where everything is so very visual, that yes, this infrastructure, that infrastructure, while parallelly we were also exploring the small microorganisms. Yes, the very small little microorganisms, and we have utilized these microorganisms also for our purpose. So here I'm very sure, because most of us are from science background. I'm not actually very sure like what is the exact background of people listening to me, but I have tried to connect it from a very basic thing to like a higher domain. So I'm just going to rush through these slides. So I'm introducing here these bacterial cells which we have utilized in, like, our day-to-day life, be it your food industry or your pharma industry and various other sectors of our life.
Now your slides are off. We cannot see your slides. I think.
Again, it is stuck, not moving again.
No, it's not visible at all. You're not sharing your screen now.
I did not change anything actually.
Okay. It got
How
disconnected.
do you want me to share this presentation, so that I will just send you this presentation? And
Yes.
this presentation is actually, the file size is pretty heavy, and it becomes a problem that even if I'm not the properties is, it is 39.2 MB. Shall I
Yeah,
just
but it
split it
will.
into parts?
No, it will come in Google Drive. If you share using Gmail, I think it will automatically come in
Gmail would
Google.
allow a maximum file size of 25 MB last time when I had presented it somewhere.
No,
So
if
I
you
will.
attach it, it will directly go into G Drive.
Okay, so let me just give it a try so that at least it goes
So
on
you
there.
can send it to my Gmail ID. I'm just giving my mail ID.
Sure.
So I've given my mail ID in chat box.
Just. It is saying Google Drive also would accept somewhere around. That is what it is saying, that not more than 25 MB. Maybe I don't know. Okay, but it's fine. Okay, it is going.
Yes.
I have just sent you, sir. Can you please have a look at it? The email ID is EV4 NextGen1.
Yeah, I received it.
Okay,
I'll
fine.
share now. Yeah.
No problem, sir. We can move forward, sir, a little bit. I think this part I had already spoken. Hello, am I audible now?
Yes, yes.
Okay, we can move a little forward, sir. I was talking about the E. coli cells. From there I'll start. One slide for this. Okay, so let me just start from here only. So I was speaking that we have a rich history of around 4 billion years of tool age, and now we are even leveling it up with machine learning and artificial intelligence. And while this is happening at a mega level where you can actually see everything happening around you, that technology has advanced to the present day inventions, but we are even utilizing small microorganisms for the betterment of human race, be it like small cells like E. coli. So I'm very, very sure that people who are from this background must have seen these images earlier also. These are small rod-shaped bacteria, E. coli, certain cocci bacteria, and of course that little one who's sitting there, viruses. So we have utilized all these organisms for our purpose. So now what advancement has happened next? These small microorganisms were used in industries like food industries, your like in brewing, in like yeast were also used in all your brewing and alcohol. All these industries were also like being catered by these microorganisms. Now what we have done is that we have moved on to the multicellular organisms. And the science today has given us this capability that we can actually extract a single cell from the multicellular organisms and then modify them. So, how is this modification done? And I think we are all aware. Sir, next slide. So, we are all familiar with this image that this is the DNA and the genetic material, the blueprint of life. So, all the microorganisms do have this genetic material, which we can edit and insert our gene of interest into these microorganisms. So, next slide. Okay, so this is how, so you can move and I will keep explaining because there is not much that I have to explain it here. So this is just go through. So basically what we are doing is that we are taking these either single cell organisms or we are taking a single cell isolated from a multicellular organisms and we modify them. And once we are modifying them, we are generating a recombinant cell and this recombinant cell is then producing what is required by us. So this brings me to this class of molecules which is known as biopharmaceuticals or biologics. And technically speaking, when we talk about biopharmaceuticals, they are basically anything which is derived from the living microorganisms. Either you derive certain active substance directly from that microorganism or you are making them to produce something for you. All that comes under this section of molecules which are known as biopharmaceuticals or biologics. So all of us who have actually prepared for medical entrance examinations would actually recall that there is an entire list of biopharmaceuticals that is there, ranging it from small clotting factors, growth factors. But what I typically am catering to is like the two-star performance from this list, which is the monoclonal antibodies and your vaccines. From this entire list, these are two-star performers, wherein that today's talk would cater about. We will be focusing on monoclonal antibodies and the vaccines. So, okay, so the reason why we will be talking about these two particular molecules is you can see in this particular distribution of like which factors, and what molecules are actually catering to the modern day requirements with respect to the therapeutic molecules. So you would see that monoclonal antibodies and vaccines will are cater to a major chunk of biopharmaceuticals, and that is why these two molecules are focus of most of the companies who are working in the sector of biologics or biopharmaceuticals. So welcome all to this world of biologics, and I'll take you through these slides how we are basically working with these microorganisms and how we are making them produce what we call as the biopharmaceuticals, biologics, or now we call it as biosimilars. So while I was discussing that there is so much around us and so much that Mother Nature has to offer to us, and we have utilized all the microorganisms, the cells, be it from multicellular, single cellular, and there is so much on our platter. What we particularly in biopharma do is this, that out of all these single cell organisms, we are particularly working on one particular bacterium, which we all know is E. coli. And after E. coli, what we do is that this is a single cell microorganism. And when we talk about the multicellular organisms in our industry, we are isolating cells from this little Chinese hamster, uh, with this little Chinese hamster, and we isolate cells from its ovary, which are known as Chinese hamster ovary cells. So when we talk about the biosimilars, biologics, these two particular organisms, E. coli and the cells from Chinese hamster ovary, are catering to a large section of therapeutic antibody production. This pie graph will actually give you an idea that how much CHO cells and bacterial cells have catered to the requirement of therapeutic antibody production. Next slide, sir. If we talk particularly, if we are talking particularly about the mammalian cells, then again this slide will tell you that CHO cells are actually able to cater to most of our requirements when it comes to the production of therapeutic molecules. There are many other cell lines to this purpose, like when we have cell lines from insects, when we have cell lines from other organs of the hamster, but still you would see that Chinese hamster ovarian cells are particularly catering to the requirement in a large volume. So basically, what are we doing? Either we are making E. coli work for us after modification, or we are making any animal cell work for us. So what are we doing? Basically, we are just modifying them, and they are working to for us like a factory. Like a factory, why I'm saying? Because yes, they are working for you and they are making for you what you are deciding. So in other words, if I have to say, I would rather say that they would work as a host, technically speaking. Factory would be a very relatable term, but then if in scientific terms I have to tell, then I would rather say that they are hosts for us. Next. So is it really that simple that, I isolate a cell or I take any microorganism, I do certain modifications in them, and then the cells are producing something that we require? But actually, we're actually very simple as it sounds due to the technological advancements that we have in our hand today, but it is actually like a treasure hunt. That is, what you want them to produce is definitely produced, but then there is also lots of other things that are present when then these cells are preparing your molecule of interest. So I welcome you all to the today's topic, which will talk about the quantification of host cell proteins as a part of process-related impurities. Myself, Dr. Deepti Gangwar, as sir has initially introduced me, I am working in analytical department where we actually cater to this requirement and we tell you that how much your molecule of interest the cell has produced, and what are the other impurities which are actually not desired by the anyone who is taking that particular molecule. Next. So host cell impurities in biopharmaceuticals, this is our topic of concern today. And what I will be talking is this: what are the types of host cell impurities? What are the various regulatory aspects which govern when we are catering to host cell impurities? The impact that these impurities have on product, what are the various analytical techniques which are used for quantification, and finally giving you certain glances about how are we developing a method for the quantification. Taking them and talking about them one by one. So I was why I am particularly taking you again and again back to a cell, because this is where we have done everything. We have modified. Say, suppose this is the cell. We have done the modification. We have changed it. Our gene of interest has entered into it, and now this cell is our host, and it is producing our molecule of interest. But then, what will happen? It, either your molecule will be inside the cell, or it will be in the milieu, which is outside the cell. So whatever be the case, now say let us take one example that my molecule of interest is in the cytoplasm of the cell. So ultimately, to get your product out of that particular cell, you will have to burst open the cell. So when you are busting a cell using any of the techniques that are available, what you are doing? You are also releasing whatever there is, that the entire machinery that the cell has, the other proteins, the DNA, the membrane. So everything would be there. So this all, which is apart from your molecule of interest, comes under host cell impurities. So in biopharma, or even in the small molecules also, when we talk about impurities, there are basically two different sections of impurities. You have the product related impurities, or you have process related impurities. I'll try and explain like one by one. So when we talk about product related, as the name itself is very suggestive, that it is something which has been generated uh, uh, from your product itself. Now when we talk about the large molecules, say a protein, the protein, when it is being produced, it can aggregate, it can fragment, or it can have several oxidations, deamidations, which can change its basic structure. So when you generate these kind of impurities, they are called as product related impurities because they are directly coming from your product. But when we talk about process related impurities, so they are all those impurities which are not from your product. They are derived from the process that you are using. So in this process, when we talk about biopharmaceuticals, what are we using? Of course, we are using cells. And when we talk about cells, so they are being used in process. So whatever will be derived from these cells will fall under the section of process related impurities. And as I mentioned in my last slide, that when you burst open a cell, you are releasing everything around them. That is the residual DNA, your host cell proteins, the endotoxins in case you're using the E. coli, and all the culture medium proteins. So these all fall into the section of process related impurities. Now, what are the regulatory bodies which go on? See, as far as when we are doing, academic research, we are more interested in how we can modify these cells and how we are actually able to make them produce what we want. But when we are taking these molecules to the market, it is not as simple as that. You have to follow certain guidelines. There are regulatory bodies sitting there which will keep a watch that your product is safe, it is effective, and like there are certain guidelines, certain aspects that your molecule have to meet before it is going into the market. So there are three like bodies which govern what are the basic guidelines, what are the aspects that your molecule has to fulfill. So these are three bodies: ICH, International Conference of Harmonization, WHO, and FDA. These bodies basically decide that what are critical quality attributes of your molecule. By critical quality attributes, I'll explain it further. So let me just go through this particular five sections. So what are the acceptable ranges of these attributes? What is the level of importance and risk, and how do we monitor and how do we control? So one by one, when we talk about these, so what are the critical quality attributes? Now, critical quality attributes are all those aspects which directly impact the safety of your molecule. So when we talk about the process related impurities and the impurities generated in by bursting open these cells, you basically what you do is this: the host cell DNA and host cell protein, as per these agencies, fall into the section of critical quality attributes. That is, they are known to directly impact the quality of your product. Then moving on to the what are the acceptable ranges. The acceptable ranges are also defined by these bodies, wherein based on their historical experience, whatever knowledge that they have gained from the biopharmaceuticals that are there in the market, they have decided certain ranges to which your molecule should fall in before they accept your molecule. Then, what is the level? Now, this is very important for us to understand why we need to control these impurities at certain level. And then how do we monitor would then cater to the what kind of analytical methods we are using to monitor these impurities. And finally, I will touch down upon certain downstream polishing steps which make us understand how do we control these impurities. So, so let us talk about the host cell proteins. So what is a host cell protein? As I just mentioned, they are all other proteins which are prepared by the host for obviously for its own survival, which are not your product related. And then what are the various methods of analysis? So there is this small list of methods which is currently catering to this particular requirement, where you have SDS page, you have ELISA, you have proteomics, Western blot analysis, bioinformatic analysis, and LC-mass based profiling. While SDS page, Western blot, bioinformatics, all these are pretty qualitative techniques which will not be able to give you an exact value of how much this impurity is present. So my talk will be focused upon the quantitative techniques out of this entire list, which is ELISA, and we'll touch down the LC-mass based profiling also. So before we actually understand the analytical methods used to quantify them, let us talk about what impact and why it is so important to control these impurities at certain levels. So basically, the impact of impurities is classified into 4 different sections. The first is the immunogenicity. The second is the proteolytic nature of the HCPs and its impact on the drug efficacy. Then how do they impact your host cell, the host cell proteins, how do they impact your molecule? And then aggregation. So let us talk about the immunogenicity of the HCP, which is a very important aspect. So I think because all of us are from a bio background and we understand what happens to us when we are under a constant fight, the pathogens are attacking our body and our immune system is responding to this particular attack. So this basic slide is just like we are continuously under attack by the immune system, and our immune system is continuously fighting with the pathogens that are around us. I have introduced this slide just for those people who have not immune system is something new to them. I was not very aware about the kind of audience I'm catering, so I've just introduced these basic slides. So basically what happens is that the moment we are under attack by certain pathogens, they try to invade us. And our first line of defense is obviously our skin, our, there's eyes, there is these secretions which control these like pathogens from attacking our body. But say what happens if they're able to attack and enter into a bloodstream. So once these molecules, these pathogens are into our bloodstream, they come across our body cells, which are red blood cells, white blood cells, and we all know white blood cells are the cells which are the defense cells. The moment they see anything that is foreign to them, they would immediately attack and engulf it and would try to clear it off from our system. So now why I'm talking about the immune system here is this, because immune system has the capability that it can differentiate between what is your body part or what is your, what is my own body cell, or what is inherent to me, and what is a foreign thing to me. So this concept is very, very important, that the immune system is able to differentiate between self and the non-self. That is, anything which is foreign to our body, our body is definitely responding against it. This becomes very important because host cell proteins are of course foreign to your body, because all therapeutic molecules, as I said, they are either being produced in E. coli or they are either being produced in certain other mammalian cells. So if you have host cell proteins as an impurity sitting along with your molecule of interest, and if you inject it into the body of a person, then obviously they will be recognized as foreign. And the moment they are recognized as foreign, your body will be triggering a response against it, and you will have a cascade of immunogenic response. And to understand this, let me bring you down to this particular case of HCP driven immunogenicity. While in COVID, most of us must have heard that there are certain blood clots that people are facing after the vaccine, like the moment they take vaccine, certain hours later, there were certain cases that yes, there were blood clots reported from certain patients. While this is happening, so this two molecules, I think two vaccines, Janssen and Janssen and AstraZeneca, they actually reported that yes, blood clots were a problem in case of these vaccines, which is in scientific terms is known as thrombocytopenia. So obviously, it was taken off shelf by FDA for a certain time, and when the research was ongoing, it was actually proved that it is not because of the main molecule of interest, that the vaccine per se is not causing any impact. It is because of the HCP that was present in that vaccine was triggering a cascade of immunogenic responses and was actually, hello.
Hello?
Hello, am I audible,
Yeah.
sir? I
Yes,
am
yes.
getting a message that you lost the network connection.
Yeah,
I am
little bit, but
not.
now it's fine.
Okay, actually, I'm not able to see the slides.
I think your network is very slow, I think.
I have changed it to another. Just hold on, sir. Okay, am I audible now?
Yes, yes.
I'm so sorry for these technical glitches.
No, it's
Okay, so while I was talking about the case of immunogenicity, which was driven by HCP, whereby these two vaccines have to be withdrawn by FDA, and till the cause was actually found out that yes, one of the host cell protein, which was present along with the main molecule of interest, was causing this immunogenicity reaction, which was leading ultimately to the blood clot. So, why I brought about this particular case study was because of the fact that this tells you that yes, host cell proteins are a part of critical quality attributes, because until unless you will not be able to tell the authority that they are present below certain levels, acceptable levels, you will not be able to qualify your molecule for the markets. And the reason is that yes, they are immunogenic, they will cause problem, they will safety, they are threat to the safety of the patient, and that is why it becomes very, very important. So moving on to the next aspect, proteolytic SCPs. So first aspect was the safety. The immunogenicity would cause the problem with the safety of the molecule. The second is the proteolytic. It's the previous slide only. So the second is the proteolytic HCPs. Now we know that any cell would contain lots of proteases, lipases, and there are other like like chopping machinery which is available for cleaving of the proteins. So say suppose one of your host cell protein has this capability that it is able to chop off the proteins. So then of course if it is present with your molecule of interest, it will carry on with its proteolytic activity and will chew off your molecule in the during the storage or during the preparation, which will ultimately impact the efficiency of your molecule. Third is the binding of host cell protein to the recombinant protein. So now you have to understand this very simple thing that all these therapeutic molecules have their active sites, right? So whereby they are known to, because of the presence of those active sites, they bind to their target molecules. Now say suppose the host cell protein that is present in along with your molecule of interest goes and just sits somewhere in that pocket, and then of course it is not available for binding to the downstream molecule, which is the actual purpose of the therapeutic molecule. So again, it will impact the efficiency of the molecule. And last but not the least, if the host cell protein itself aggregates further, then it can again trigger the immunogenic response, which will then impact the safety of your molecule. So these are the four basic aspects why HCPs are important and they have to be controlled at certain levels. So if they are so important, how do we actually monitor and how do we remove them? So this is just to give you a gist that this is a downstream processing whereby we remove the host cell proteins from your molecule of interest in a sequential manner. To understand this particular slide, we all know that when we have modified a cell, we will then raise it to large volumes in a reactor, bioreactor, which is you raise it to 100 liters, 1,000 liters, because obviously a single cell won't be able to cater to the requirement. So you raise these cells in the large numbers. And when you're raising this cells in large numbers, what you then do is that you open up all these cells to release your molecule of interest, and then through a series of steps which fall under downstream processing in biotech industry, you are separating your molecule of interest ultimately from these host cell proteins. While we do this, we are sequentially removing the host cell proteins and enriching your molecule of interest, but it is not always possible to remove all these impurities. So then ultimately some of these impurities are still available along with your final molecule of interest. So then how do we actually quantify it? So this brings me to those analytical methods which are used to quantify. But the current method and the most important, most acceptable by the regulatory authorities is the ELISA, which is enzyme-linked immunosorbent assay, and all of you must be familiar about this particular technique. So yes, this list I had initially mentioned that these are the list of the methods that we are used for, qualitative and quantification of HCP. So I will talk about ELISA from here onwards because we are more interested in the quantification rather than just qualitatively estimating the host cell proteins in the molecule. So next. So enzyme-linked immunosorbent assay, I'm very, very sure that most of you have, done it or have at least read about it. So it's a very simple procedure wherein you are coating your molecule of, interest on a simple 96 well plate, and then you do a blocking. Blocking would, block all other available sites in that plate. Then you would do a detection using certain antibodies and then read the result. So this is a simple step-by-step process what you do in ELISA. I will explain it in further slides. So yes, ELISA is basically of 4 different types: direct, indirect ELISA, sandwich ELISA, and competitive ELISA. So as the slide is very, very self-explanatory, indirect ELISA, you would detect your molecule of interest by a single antibody. In indirect ELISA, you basically bind another antibody on top of the first antibody. So this becomes indirect, and the sandwich ELISA is where your antigen is in between, and you coat one antigen on your plate, then you bind your molecule antigen onto it, and then you have another antibody which is then used for detection. So this is the sandwich ELISA, and then you have a competitive ELISA. Now, when we talk about the host cell protein determination, we are using sandwich ELISA for the detection. So in this case, what you do? So you have all these host cell proteins and you have antibodies against these host cell proteins. How do we make it? We can, I will just show you in my coming slides. But what you do is this, that you are in your 96 well plate, you first coat your antibodies against your host cell proteins. And now you take your sample, whichever, I mean, your drug substance or your, finally what you have purified, that particular formulation. You can call it as a drug product or drug substance. So you ultimately take that particular sample and you just float that sample into this particular well, which is already coated with anti-HCP antibodies. Now what you do is that you coat your slides with another antibody, which is which will again detect these antigens which are bound onto your plate, and then you do a detection. So this is the format for sandwich ELISA for HCP quantification, wherein your antigen is in between the two antibodies, and that is where it is known as a sandwich ELISA. So this particular format is being used for HCP quantification. Now, how do we do it? How do we generate the reagents for this? So I think all of us are aware that this is a very crucial interaction for ELISA, that you have your antigen, and on antigen you have your antibodies which are bound onto the specific sites. Now, when we talk about this particular interaction, I'm very, very sure all of us are pretty aware that these antibodies are very highly specific, and they will be only reactive to one particular class of molecules which are known as antigens. And these antigen and antibody complexes are very, very like your lock and key hypothesis or your enzyme substrate complex. They're very, very specific. One antibody will always be able to bind to one antigen. It will, unless there is a certain degree of like matching between your antigen with respect to its sequence and its nature, the antibodies won't bind it to each other. So you have two class of reagents. One is the polyclonal antibody and the another is a monoclonal antibody. And as you can see, on one particular antigen, if you have multiple sites and you have antibodies against all those sites, then your antibody pool will be known as a polyclonal antibody. And if you have only a single antibody against one particular site, then it will be known as a monoclonal antibody. And as I just said, that these complexes are very, very specific, that you can only bind one particular antibody to one particular site. This is important in this particular case because when we are generating the reagents for ELISA, this particular interaction will play a key role. So then how do we generate? As I just mentioned, that what you do in sandwich ELISA for HCP quantification is that you coat your plate with anti-HCP antibodies. But then how do you generate anti-HCP bodies? So you remember we spoke about the bioreactor from which we are getting our molecule of interest. So when we are isolating our molecule of interest from the rest of host cell proteins, so what we are doing in this case now to generate the reagent, we will take all the host cell proteins now. Your molecule of interest is separated, but now to generate the reagents for doing the ELISA, we are isolating all the host cell proteins. Now we will raise up these host cell proteins and we will use these host cell proteins as antigens, and we will immunize goats and rabbits using these antigens. So you remember, as goats and antibodies, we are producing antibodies in these animals, and now this is an E. coli culture, so it is of course foreign to them, so they will also start producing antibodies against these particular proteins. So now you have your large different kind of pool of antibodies against all these antigens, which is known as the polyclonal pool of antibodies against all your host cell proteins. So this is a very critical reagent, and this is produced and is required, that usually regulatory bodies would accept and would want that you always do and follow this particular method for generating the reagents for doing your ELISA quantification. So I'll just explain the slide again. You raise up the culture in bioreactor and you are separating all the host cell proteins now, and you use these host cell proteins to immunize different animals, say goats and rabbits. And once they respond towards these antigens, you separate all those antibodies from their serum, and then you qualify that as a reagent that yes, these are responding to my these host cell proteins, and then you use this as a reagent. If anywhere there is a question, please ask me at the end of the session. I would be happy to answer. So now we have your the entire pool. There will be many antibodies and lots of antibodies against all of your proteins. Then it becomes a polyclonal pool of antibodies. So this becomes a reagent for your sandwich ELISA. So now you use these antibodies from one animal, say goat. I have uh, immunized goat and I have generated one set of antibodies, and now you have rabbit also, which are immunized with the same anti-HCP, these HCPs, and they are also producing your anti-HCP antibodies. So you have two sets, one in goat and one in now rabbit. So now what you do next? From one particular animal, you will do the coating, and the other animal, you will do the detection. This is the format of the sandwich ELISA that we are using in the HCP quantification. So next slide. So this is the flow chart that you take the total HCP extract, you do the immunization of one set of animal, and then you do the immunization of another set of animal, and you isolate all the and purify and all these anti-HCP body, and you generate a pool of anti-HCP body which are then qualify as your final reagent. So once you have done all this, the most important part is this, that you have to check the antibody coverage. Now, when I say that, there are, say, suppose 100 HCPs or 100 E. coli host cell proteins which are present in my drug substance, but will they be all immunogenic? The answer is no. So, out of, say, suppose 100 proteins that are present in your pool, maybe say suppose 70 to 80% are will be those proteins which will be able to trigger the immune response in a rabbit or in a goat. So by that token, it is not always possible that whatever pool you are injecting in these animals, all of them will not be able to generate the antibodies. So by this token, when we talk about reagent generation in terms of ELISA, you have to actually show that if I have injected 100 proteins in my goats and rabbits, what number of antibodies I am able to get out of those particular animals. And you have to actually tell that out of 100, either 70, or out of 100, either 80 are detected, and that is where you have to show that my critical reagent is able to detect only 70 or only 80% of the entire pool of host cell protein. So this becomes a very, very critical reagent, and while doing this, while presenting it to the regulatory agencies, this is very important that you are actually telling what percentage coverage of anti-HCP antibodies you have generated. But what is the problem in this particular case? The entire problem is this, that right from raising the antigen to animal immunization, and then when you qualify these reagents, this is a very, very long process. When we have generated it for one of our product, the usual time that the agencies would take is usually around 2 years. Yes, you heard it right. It is around 2 years that you take to for this entire process of isolating the host cell proteins, then immunizing, then qualifying the reagents, and then ultimately you are using these particular reagents. So what is the answer to this question? So while we are developing the product, it is not very possible that every time you do this process. So there are certain generic kits which are already available in the market, which would tell you that, say, suppose you are using E. coli or you are using a CHO cell, you can use this particular kit, and this kit is able to cater to this much of your E. coli. So they will also tell you that this is the coverage and this caters to this particular strain. So while you are developing your product, this is product specific. This product specific reagent generation you can take up later on. You can meanwhile actually use the generic kits which are already available in the market, and parallelly, while you are developing your product, you can develop these reagents, and later half when you have like developed your product and it is has reached to a certain level, you can use product specific antibodies, and then you can replace your generic kits which are you have used during the initial phases of your method development, and then you can replace it with the product specific antibodies. So this is like a general kit that that we use for quantification, where they will already provide you a pool of anti-HCP antibodies. Then obviously they will provide you these standards, these small bottles that you are able to see. These are the small bottles of pure HCP. Now, in all of your analytical techniques, wherever you have seen or you have worked, you always need to have a standard curve. Now, when we talk about a standard curve is usually prepared using a pure material. So what these small bottles are, that they will provide you host cell proteins in a known quantity. Either it will be 1 nanogram, 3 nanogram, 5 nanogram, 10 nanogram. So these are small bottles of a known quantity of host cell protein. So the concept is very simple, that you take these known amount of host cell proteins, you plot a curve using these standards, and then you quantify your material against these particular standards. So what is the current regulatory expectation? Regulatory expectation is, as I just said, as I initially mentioned, that being a part of critical quality attribute, you have to always show that what is the level of antibody coverage that your antibody is able to do. And then there are certain limitations of ELISA. As I said, that 20 to 30% of your HCPs will not be detected by ELISA. So regulatories have an expectation that you will always fulfill it up with certain orthogonal methods. So ELISA, because of its the once you have the reagents in place, ELISA is a very high throughput technique. You can do it, lots of samples in a single go. But then, because of certain limitations of this particular technique, there is always an expectation that you are providing certain additional data, or additional methodologies are being used to do this quantification. So currently, as on date, we have LC-mass based profiling also, which is being done in certain cases, but it is only and when there is certain cases of immunogenicity, that that this particular molecule is known to cause immunogenicity, and I have to quantify it to the actual levels. We all understand that LC-mass is a very accurate technique. So that is where, in case of any regulatory expectation, we have to support it by certain other techniques, which is LC-mass in the current case. So while with respect to the HCP reagents, as I just mentioned, that for ELISA reagents, you have to show the coverage, you have to tell what range of molecules, what molecular weight of HCPs my particular reagent is covering, high molecular weight to low molecular weight, and the degree of coverage is very important to qualify your reagent. But you can always parallelly support your particular product by using certain other orthogonal techniques to tell that how product what is the level of purity of your product. So now moving on, talking about LC-mass based approach of HCP profiling. So I am not a very LC-mass person because I have never done it myself, but I just wanted to give you a glimpse of this particular method, that you have all your HCPs in your sample. So basically you do a fragmentation. Fragmentation would generate your peptides, and then the peptides will be matched to the libraries, and then you can identify even certain the accuracy, the sensitivity of this particular technique is very high. But then the entire problem is that the moment you change anything in your process, the moment there is anything you change even in your substrate, it impacts what kind of host cell proteins would then be finally available in your final product. So LC-mass technique is obviously by far a very highly sensitive and accurate technique with respect to ELISA. But when it comes to its applicability, so it becomes restricted in terms of this fact that any slight change in your downstream steps, any slight change in your substrate would impact the analysis. So LC-mass, of course, is a technique, but then it has again its certain limitations, that you would need a very, the manpower, the kind of manpower that is required to do this kind of quantification. That is also trained manpower, of course, is required. And then the moment in case of any changes, the technique would change, the kind of profiling you would get would change. And then when it comes to the throughput, being in throughput, so obviously LC mass is not able to match ELISA in terms of being throughput, high throughput. So as I just mentioned that even as on current date, with respect to HCP quantification, ELISA is still considered as a gold standard technique. And so to develop a method of ELISA, we have to follow certain rules. So number one, as I mentioned, that you have your HCP analysis, you have to show the antibody coverage. Then what is the dynamic range of your standard? The determination of minimum required dilution, the analysis of matrix interference, and evaluation of spike recovery. So all these factors, once you are developing a method for your HCP quantification by ELISA, you have to take care of all these parameters once you nail down your final method. So let us take and talk one by one. HCP ELISA coverage, as I just mentioned, that when you are generating a reagent for ELISA, so basically what you have to ultimately show that if at all there were 100 proteins present in my initial pool of host cell proteins, and after immunization, how many of these how many of these proteins are giving me the antibody response, and finally in my pool of antibodies, if at all out of these 100 proteins, how many of them are actually recognized. So that is the coverage. So to show this coverage, what you do is that you run a two-dimensional SDS page. Two-dimensional SDS page, I'm sure people must have seen. So you run two SDS page parallelly, and out of which one of them you do a silver staining and the other you do a Western blotting using the anti-HCP antibody reagents that you have generated. So now you do a computative analysis where you match spot by spot, and that will give you the exact coverage that out of total spots that are detected on a silver stain, how many spots are being detected on your Western blot. That would tell you that what is the percentage coverage of my particular reagent. So to begin with any ELISA for HCP quantification, this is the first requirement. Now talking about the dynamic range of standard. So because we are doing the quantification, it is obviously very, very necessary for you to show that the standards that I'm using are falling in a linear range of that particular assay. For all of you who have actually done these kind of assays, you would always remember that it is always best that you are working in the linear range of any assay. Anyway, when you will go towards the lower side or the higher side of assay, would either you will lose your sensitivity, or you will not be able to actually quantify towards the higher level because of the saturation or due to the limitations of certain reagents. So you have to always show in what dynamic range is my standard functional. That is, because see, it is not that we are dealing in NGs of certain samples. We are actually, when we talk about HCP, so they are they are present in your sample at a level of ppm. So we are talking about something which is present at a very low level. So it is always very, very important in what dynamic range you are working will obviously tell you. This will actually also tell you to what dilutions you have to do for your samples. This I will explain in further slide. But dynamic range of the standard is crucial, so that dynamic range of the sample is crucial so that you can actually establish what levels I will be able to work with, so that I'm actually able to quantify my host cell proteins very, very accurately. Now, the next important point is minimum required dilution. So, if there are so many like people in the audience and they are still actively listening to what I'm saying, I would want to know that is it possible that you can dilute a sample endlessly to a point? Say, suppose most of your bioformulations are in a range of 100 mg per ml to 25 mg per ml to 100 mg per ml. So, I mean, can anyone answer to this question that, as I just mentioned, that you have a dynamic range of your standard and now you have a sample. Now, my dynamic range of standard is dealing somewhere in a ppm, and my sample is at a level of 25 mg per ml to 100 mg per ml. So, can I dilute a sample endlessly, or if I can, take my sample as such? Anyone in the audience can answer to this question that what should I do and how I should start? Anyone? Okay. So let me explain this to you. So as I just mentioned that we are dealing with the host cell proteins which might be present in your sample at a very fairly less levels in some picograms or nanograms. So when we are talking about our sample in which we are detecting these HCPs, so we have to dilute our samples. Dilute our samples in such a manner that they fall in the dynamic range of your standard. We are talking about ppms and our sample is in the range of 25. We can use the sample as such also, but just imagine your sample, your molecule of interest is present at a level of 25 or 100 mg per ml, whereas what you are detecting is present in the level of picograms, nanograms, like at this level. So obviously, the more the amount of your molecule of interest in that particular matrix, in that particular sample pool, it can hinder the actual detection of your HCPs. So what is that then you do? So basically, what you do is this: that you start diluting. You start diluting your sample to certain level that it then comes and fits into that range, and that is known as the minimum required dilution. Obviously, I can't dilute my sample to a range that it is, now the range has fallen and gone down below the lowest standard. Obviously, I can't do that also. So I have to somehow figure out what will be the dilution of my sample, known as minimum required dilution, which can actually accurately quantify my CPs. So how do you do it? So you start diluting. Sir, can you go to the next slide because that will explain. So you have your starting sample, stock sample. So now you prepare a serial dilution. Serial dilution is like, the way you prepare it for all other techniques. So you have started preparing a serial dilution just to understand which is that dilution which is optimal for my assay. So now you, in the initial of your experiments, you analyze all these samples. So when you have all these samples analyzed in your particular standard curve, some of these samples will be below your last standard and some of these samples will be above your uppermost standard. Only few of them in the middle of this dilution will fall into your range of your standard. So now out of all, say suppose 3 of these dilutions fall into your range of standard. So even from those 3, you have to select which is the dilution, which is the optimal dilution for this particular assay. So how do you do it? Say suppose I talk about 75, 37 and 18.8 pg per ml. These 3 concentrations are now falling in my range of dynamic range of my standard. So all of them, when corrected for their dilutions, will give you somewhere a close value of your HCP. Say suppose 75 after calculation is giving you X ppm, then 37.5 should give you 0.95x, and this 18.8 might give you 0.80x, but they will always be somewhere correlating in the range of 80 to 120% of your X. So that means there is one concentration that I have reached where the values of my HCPs that I am getting is within a range. So out of these 3 concentrations, then you decide that let me work with these 3 concentrations, which is giving me more or less a fixed value of my host cell protein. So this is how you decide that these 3 concentrations which are falling within my dynamic range of my standard are giving me somewhere a close value of HCP. These 3 values are those values or those dilutions with which I will be working forward. Now to nail down out of these 3 samples, what will be that concentration which is giving me the most accurate result? This is how you then move to the next step of your development. So previous slide, previous slide. So I told you minimum required dilution, that you are getting a value, a constant value in the range of 80 to 120% of your expectation, which is X. Now out of those 3 supposed concentrations, we move on to which is the best or the optimal concentration of my sample. So you do a spike recovery analysis. Now what is spike recovery? You can read this line, but let me just explain you. Spike recovery is suppose now, as I told you, that you diluted certain your samples to a certain level and they have an invariant value X. Now what you do is this, that you add suppose Y into all of these three samples. So ultimately, after doing your analysis, you should get X plus Y in these samples in which you have spiked. Again, I'm explaining. You have three concentrations. They have certain levels of endogenous HCPs, and when you spike all these samples with, say, suppose Y is again an HCP. So you should get X plus Y. So if you are able to generate your answers that X plus Y is what I am getting out of these three concentrations, this is the concentration which is giving me most accurate X plus Y. It will be the concentration of the sample that you will be diluting and you will be taking for your assay. So this is basically a spike recovery assay which you are doing for proving that your ELISAs are accurate or they are accurately quantifying your protein. So you do a dilution series, then you do a spike recovery assay just to show that these samples or the concentration that I'm taking and the way when I have spiked them with certain known amount of an analyte, then my assays are able to give me the exact or at least a closer value of what is my theoretical concentration. So once you show that you have these concentrations which are giving the spike recovery value, so then you can say that my ELISA is working accurately, it is working perfectly. So this is how you initially start. So you fix up a concentration of the sample that will be used, you spike a known amount of SCPs in that, and then you do a ELISA, and then you show that yes, I am getting the spike recovery values. So next. So this is what I told you about the minimum required dilution, that you do a dilution. And then when you get a constant value, that is between those concentrations, you are able to finalize, say 2 to 3 concentrations in which you do a spike recovery analysis. So next slide. So this is what I have my sample which has certain endogenous amount of HCPs present in it. Then externally you spike certain additional HCPs into it. Now you know that you are expecting X into it and you are expecting Y also in your spike sample. So your method should give you anywhere a value between 80 to 120% of your X plus Y. That is the analysis which is accurate and as considered as good. The ELISAs, when they fall in the range of 80 to 120% of your expected values, are considered as good ELISAs. Next. What if this doesn't happen? Say I'm not able to get a recovery between 80 to 120%, then what am I supposed to do next? Then first in that case, I will have to understand that like what could be the potential interferences. Now potential interferences could be due to your buffers, it could be due to your another antibody which could be present in your sample, or your own molecule of interest which is not able to which is somewhere interfering with the actual quantification of your HCPs. So what you have to do is this: either you have to again say, suppose out of these three concentrations the highest concentration is not giving me the accurate value, then I will go down to the second the middle concentration. So then I will keep working with these two concentrations. So by that token, what I mean to say, if you know that it is because of my sample, then dilutions would provide you an answer that yes, I can dilute my samples to, say, suppose the next possible dilution, and that is the way I will avoid the matrix interference. Anything which is present in matrix and it is impacting the accurate estimation of your HCPs, that is known as the matrix interference, which you can solve by the fact that you can dilute your matrix. So that is how you cater to the problem of matrix. And in case if it is due to certain buffers which is present and it is incompatible, then you can do your buffer exchanges. So that way you can just say, suppose I have traces and I have certain urea in my drug substance which can actually hinder the protein interaction. So then in that case I will do a simple buffer exchange, so that in the downstream processing, when the antigen and the antibody reactions are happening in my plate, they are not hindered by these components. So basically in ELISAs you would encounter these kind of problems which can be easily catered to. So this is all about how we develop ELISAs and how we actually quantify our ELISAs. So doing a summarization of all that we have gone through today is that we actually understood there are two basic class of cells that we are using in biopharmaceuticals, largely E. coli and CHO cells. Then we understood what kind of like impurities they generate in biopharmaceuticals. The cells, because they are related in the process, so the impurities that we generate from the cells are fall under the section of process-related impurities. Then we understood why we have to, take care of these impurities. The fact that these can be immunogenic to you, they can impact the safety of the patient, or they can actually impact the efficiency of your molecule of interest. So that is why it is important that you keep a check that what are the level of these impurities. Then what is actually, host cell protein and their direct impact on therapeutic molecules. That is what we understood. And then finally, what is like analytical methods, out of which we talked about ELISAs and HCP, LC-mass based HCP profiling. And then what are the critical reagents that we need to do ELISA, and finally, how do we actually develop our ELISA method in our lab. So this is what I tried covering in this particular session, and I'm extremely sorry for all those technical glitches. I wanted to deliver a wonderful presentation. Hopefully I did some justice to you guys, and if you have any questions to me relating to this, then you do drop me a mail at this particular email address. Thank you. I'm open to the questions now.
Yeah, thank you very much, Dr. Deepti, for a wonderful talk. But in between, we had few glitches, but still we could understand about how the HCPs are determined and their